Hi everyone. Hope you're doing as well as possible while at home. Just want to say that I miss seeing you all every day, and I'm here for you if you need anything. This may be one of the last sessions that you see me since I'm going on maternity leave soon, but I will. I'll post a little goodbye video maybe later in the week and keep you posted on that. But in the meantime, we are going to attempt to do some note taking with the use of this screencast and PowerPoint. So I'm going to walk through this with you. And again, please reach out with any questions or anything at all that you have or any issues. I'm here for you, and so is Mrs. Ferguson. Okay. So we are starting on Unit 5, which is Chapter 10. So we're going to start by talking about the cell cycle, basically the life cycle of your cells. So let me just get this started. Okay. So mitosis is basically nuclear division, meaning the division of the nucleus of a cell, which then results in one cell becoming two cells eventually, right? So basically this overall process is where you start with an original, what we call a parent cell, and that parent cell will divide to become two daughter cells that are identical to the original parent cell, right? So what I want you to think about with this process is why cells might divide, right? So let's say you get a cut on your finger, right? You need to produce new skin cells for that finger to heal, for that cut to heal. The way that your skin cells do that is by your skin cells that exist there. They actually multiply by dividing, right? So basically they make identical copies of themselves, which is what we're talking about here. Now, again, so why divide? So that's for repair. So your body needs to repair itself or replace cells. And also, this is the way that you grow and develop. So you started as an original, just a fertilized egg, right? So you were a sperm that fertilized an egg. You were one cell. So the way that you became many cells is by your cells dividing and multiplying, right? Making identical copies of themselves, and eventually becoming an embryo, and then a fetus, and then you, right? So prokaryotes, let's start with just a little review of prokaryotic cells. So hopefully you remember that prokaryotes are bacteria. So these are much simpler cells, right? They don't have a lot of stuff going on inside. They just have DNA and a few other things like ribosomes to make proteins. So when prokaryotes, which are, again, just a category for bacteria, when they divide to reproduce, they actually divide by what we call binary fission, which is very similar to mitosis. So basically you have an original bacterial cell, and all it's going to do is make a copy of its DNA and then pinch in half, like that, right? So we call this asexual reproduction because the bacterial cell is making an identical copy of itself, and there's only one parent involved, so no partner, right? So basically, when you make clones through asexual reproduction, that's again one parent just making a copy, an identical copy of itself. So that's asexual reproduction. One parent, and then identical offspring. Eukaryotic cells go through mitosis. We do not go through binary fission like prokaryotes do, although our process is similar. So basically, what our cells do when we need to make new cells, again, for growth, for repair, for replacement, right? We go through mitosis. So this is basically the process where you have an original cell, and that cell is going to make an identical copy of itself. And so your result is going to be two daughter cells that are identical to each other and identical to the original parent cell. So just to do a little review of chromosomes. So chromosome is your DNA, right? It's just a term for DNA, and it's basically the form your DNA takes when it gets super organized, and you're probably familiar because chromosomes look like X's. So a lot of times when we see pictures of DNA, we'll see pictures of X's. So that's basically your DNA when it's super condensed in order to get organized so it can separate and make sure that you have DNA in each of your resulting cells, right? So the chromosomes carry your genetic information, and each organism has a specific number of chromosomes. So we have 46 chromosomes total, or 23 pairs. And we say 23 pairs because you get one set of chromosomes from your mom and one set from your dad. So your 46 chromosomes, you're basically half of your genetic information is from mom and half is from dad, right? So 23 pairs or 46 total. But any living thing, again, has DNA and is going to have chromosomes. So those numbers of chromosomes differ depending on the organism. So carrots have 18, for example. Fruit flies have 8. I think dogs have like 78. So we do not have the most chromosomes, even though we're the most, right, we might consider ourselves the most evolved, or the highest functioning. Doesn't mean that that's because we have the most chromosomes, because we don't. So here's just a diagram of chromatin versus chromosomes. Again, these two terms are referencing DNA. So chromatin and chromosomes, they're both forms of DNA, right? And again, your body has DNA. Your DNA holds the instructions for all of your traits, right? Because your DNA codes for which proteins your body will make. So when we look at these two diagrams, here you can see on the left, you have chromosome, again, that famous X structure. This is when DNA is super organized, and it coils up, right? Now, DNA, when it's in its format that it takes on a daily basis, is going to be more in the form of chromatin, where it's less organized and jumbled like spaghetti. But again, when your cells are getting ready to divide, you need to make sure that DNA that's copied, you get exact copies into each of your subsequent daughter cells. So then we take the format of a chromosome, where that organization is more clear. So here, for example, and again, we're going to talk through the cell cycle and what this actually means, but these are what your chromosomes look like typically. The S phase of the cell cycle is where your DNA gets copied. So this single chromosome, we've actually copied here. So this is one, let's say this is the original chromosome, and this one here on the left-hand side is the copied chromosome. So now we actually have two what we call sister chromatids. They're twins, right? So we have the same genetic information, just copied. And the reason for that is because we're preparing for the cell to divide, and we need to make sure any daughter cells, any new cells that we make, have the same exact genetic information as your original cells. So again, now you have copies, right? So this is your original chromosome, and this is a copied chromosome. We call them sister chromatids, right? So then after mitosis, which is this process of nuclear and cellular division, you're going to end up with two daughter chromosomes that were identical to your original chromosome, and each of these chromosomes would be in separate daughter cells. So what is the cell cycle? So this is a series of events, again, that your cells go through as they grow and divide. So this is the life cycle of your cells. So the cell spends the majority of its life getting ready to divide. So it actually isn't, your cells aren't dividing constantly. And again, it depends on the types of cells we're talking about. Some cells will divide more frequently than others. You can probably think about things that maybe have to grow really quickly or often, like your skin cells or your hair cells. Those are going to divide more often than like your liver cells, for example, right? But again, each cell spends the majority of its life getting ready to divide. So division is just a small portion of a cell's life. Most of the cell's life, it's going about its daily activities and it's preparing to divide, right? So when we talk about the cell preparing to divide, that phase is called interphase, and it has three subphases, which are G1, S, and G2, which we'll go through. Then the actual division process we call mitosis, or the mitotic phase, which we can just abbreviate as M. So this is an actual division phase, and again, we're going to go through each of these steps, which we abbreviate as PMAT, which will help you remember them, okay? Just a couple other terms for you to be familiar with. G0, this means that the cell is actually not actively in the process of preparing to divide, so it's kind of like in a resting phase. These would be like your nerve cells, because your nerve cells, when they mature, they actually don't make copies of themselves, which is why brain damage is so long lasting. Your brain actually can't replenish itself by regrowing new brain cells, because most of your nerve cells actually don't go through this division process once they're mature. Then cancer, on the other hand, is something where basically it's when your cell cycle is out of control, right? If your cells are dividing uncontrollably, then you might get too much multiplication or rapid development, and that can lead to tumors and cancer. Okay, so interphase. So again, the cell spends the majority of its life in interphase. Probably 90% of each of your cell's lives are spent in interphase, which is this phase where we're preparing to divide, but we're not actually dividing, right? So the three steps we have here are first G1. So these actually refer to gap phases, but I'd like you to think of them as growth phases, because what happens in our G phases is basically the cell grows, right? So at the very beginning of the cell's life, it enters G1. So the first thing when your body produces new cells, the first thing those cells are going to do is they're going to grow a little bit, right? So you have growth phase, and it's going to increase in size, and it's also going to do its daily activities here, your cells. So basically getting bigger, and doing its daily job, making proteins and stuff. Then in interphase, you have a second step. So this is really starting to prepare more for that division. So the S phase stands for the synthesis phase. If you remember, to synthesize means to make. So this is where your DNA is actually copied. This is super important because, again, if we're trying to make two daughter cells that are identical to an original parent cell, then you need to have identical genetic information in each of those resulting daughter cells, right? So we need to make copies of our DNA here in advance, so when we actually divide, we have DNA that's identical in each of our resulting daughter cells. All right. Your G2 phase is the final step before the cell actually starts to divide, right? So again, this is another growth phase, and this is final preparations for the cell getting ready to divide. So it's going to get a little bigger. It might make copies of organelles like mitochondria and your smooth ER and your rough ER, stuff like that. Make additional proteins. This is all getting ready to enter cell division. Okay, so now let's go through the actual steps of mitosis. So again, when we talk about mitosis, hopefully we remember that DNA is contained in eukaryotic cells. DNA is contained within the cell nucleus, right? So the nucleus holds your DNA, holds your genetic information. So what we're going to need to do in order to make a copy of this original cell here is we're going to actually need to copy the nucleus of that cell. And thus, that's why we needed to copy the DNA, then we'll copy the nucleus, and then we'll separate one cell into two, right? So prophase is basically the idea that your nucleus is going to need to temporarily disintegrate so we can get these chromosomes organized and separate them out, right? So prophase is your longest phase of mitosis, or this nuclear division. So what happens here is chromatin, which again is DNA when it's loose and it's jumbled like spaghetti. Basically here, your DNA is going to condense into this X format, this chromosome format. So it's already been copied at this point, but now it's gotten super organized in order to actually be able to be separated, right? So chromosomes condense, and they show up. Also, we have these centrioles, which we learned about way back in Unit 1, but we didn't learn much about them. So they're going to be involved in cell division, and they're found only in animal cells. So here we see these centrioles are going to actually start migrating to the poles of the cells. So they're going to actually start moving here to the poles because, again, they're actually going to help to separate out our DNA. We also have spindle fibers that develop. So these fibers here are what we call spindle fibers. So you can see they're sprouting from the centriole area. These are actually going to attach to our chromosomes in order to separate out the DNA. And then also you have your nuclear envelope, which is your nuclear membrane. That's going to start disappearing, right? Your nucleolus, which is this darker region here, that's also going to temporarily disappear. We don't need either of those for this process. We need them to go away so we can separate out our DNA. Our second step of cell division, so it is metaphase. So again, we're going through PMAT now. So the first step is prophase, second step is metaphase. So here we have our chromosomes lining up in the middle of the cell. So hopefully you remember that our DNA has already been copied. So here is one chromosome, and on this other side is an identical sister chromatid, right? So this is one sister chromatid and this is another. They're twins because the DNA has been copied. So we actually want to separate these out and pull one sister chromatid to this side, the other sister chromatid to this side, in order to ensure that you have identical genetic information that ends up in each of your resulting daughter cells, right? So what you need to note for metaphase is that your chromosomes are lining up in the middle of the cell, right? And spindles, those spindle fibers that we formed, are actually going to attach to the centromere of the chromosomes. The centromere is the center region of the chromosome, okay? So spindle fiber will attach here. It's like a landing pad, so it can basically pull the twin chromatids that were copied apart to opposite sides in the next step. So the next step here is, again, your centromeres are going to split. So you can see those spindle fibers are pulling the chromosomes. the sister chromatids to opposite sides here. So anaphase, I want you to think away. So